Cool. So I'll go ahead and get started. You guys can keep typing away at your mahia if you haven't quite finished it yet. But we are thinking about what we learned last week, what is our goal for this week, and also thinking about our random question, which is, would you rather be too hot or too cold? So let's have our little korero. So the first thing, I'll pick someone randomly to answer the question about would you rather be too hot or too cold? Person answering that today is going to be Cohen. Would you prefer to be too hot or too cold?
Too cold.
Too cold? Why? You don't know, you just prefer to be too cold. Anybody else? Hands up who chose too cold. Oh, a little bit more than half. Who chose they'd rather be too hot? Oh, interesting. Only a handful that want to be too hot. Who wants to explain their reasoning to why they want to be too, like they prefer being too cold? Jessica?
Because you can get warm easily.
You can get warm easily. That is exactly my same point. Like I could just easily layer if I get too cold. Whereas I find too hot, it's really hard to cool down without like some sort of air conditioning or something. Even a fan, I don't think really does it. All right, next question is micro goal for the week. Person sharing that today is going to be Savannah. Yes, what's your little micro goal for this week? To finish your assessments? Very good goal, I would say. Yeah, it's the last week of term, so there's probably quite a few assessments that are going around. The good news is this biology one isn't going to be due until next term, or we won't assess it till next term, so still have plenty of time to work on it. All right, anything else that anyone else wants to share before we move on to our learning tasks? No? We good? All right. So let's switch gears and think about what we are doing this week on our little learning journey. So last week, we talked about cell structure and we started talking about cellular transport, which is going to be really important for today's experiment, which is looking at a dialysis bag and exploring osmosis. Now, we haven't had a chance to talk about osmosis in great detail, but that's okay because we can use this experiment as a jumping ground to learn about osmosis, which is a form of diffusion. Really, really important because this is going to be part of the context of the internal that we will do. So we're going to be doing two key experiments to help you guys prepare for the internal that you'll do next term. The first experiment is on osmosis with this dialysis bag, and the second one is going to be on surface area, looking at some pheno jelly. So both of that is really important, and basically over this whole week we will work on this experiment and analyzing it. Does that sound good? All right. So, last thing we need to do before we start is our weekly fakatoki. So this week we have he ihu kuri he tangata hare, which is as a dog follows a scent, a traveler relies on hospitality of others. The other way you can think about this is beware blindly following your nose, be deliberate and make good choices. And I'd say that's a very good fakatoki for this week, considering it's the last week of term and you guys are probably going to really need to be focused to get those assessments done. Cool. So here is today's lesson plan. The first thing that we're going to do is Mr. Hall is going to introduce himself because he hasn't had a chance to do that. Then I will show you guys a video on how to set up your dialysis bags. We'll talk about the experimental instructions and then you guys will set up your dialysis experiment. Depending how we go for time, we might have a chance to also talk about osmosis and what to expect with that. Looking at a video and also talking about it in relation to our mind map. And Mr. Hall will be covering those last two bullet points. Sound good? All right, last thing I want to do is talk about the checklist, which I have finally gotten around to making. So I'm sorry if our previous numbers don't match up. The front bit of the checklist has everything that we're covering content-wise, and the back bit talks about the assessment criteria when it comes to this internal. Remember for this internal that you will be doing some of it together and some of it alone. So basically the experimental component of it, you're going to be getting to do together. For today, we are still working on that cellular transport, and we're going to be practicing the planning the experiment section, which covers number 4 through 8 on that success criteria, just so you guys are aware of what we're working towards. Like I said, it's also on the back that's more specific to the assessment itself, so you can see how the Achieve Merit Excellence breaks down, what you're going to be allowed to do as in your pair, and what you have to do on your own. Cool. All right. So I will now pass over to Mr. Hall to give him a chance to introduce himself, and then we'll move on to the experiment.
Alright, kia ora koutou katoa, ko rangitoto te moanga, ko tikapa moana, ko Ngāti Pākehā te iwi, ko Mr. Hall toko ingoa, no Tamaki Makaurau e noho nei o, no reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. So yes, I am Mr. Hall, and I am here at Edgewater as a student teacher. So I'm supposed to be here for 9 weeks, but I haven't seen you guys for a couple of weeks because I had COVID, but I'm back now and I should be here for another 6 weeks. Just a bit about me. I was born in Auckland and I spent basically my whole life here except for when I went down to Otago to study. So when I went down there, I studied food and nutrition. So that's quite a lot of my background. That was really my passion. When I was at high school, bio was actually my favorite subject, not even lying. So I do really like the subject. And if anyone ever has any questions, I will try my best to answer them. If I can't, then I'll find out for you. So I'm here for another 6 weeks, so you're probably going to be hearing my voice quite a bit more. Otherwise I'm just excited to be here.
Thank you for that. All right. Shall I move on? Talk about dialysis. So, with this experiment, we are trying to think what's the best way to show you guys what we're doing. Maybe we will go to the dialysis bags, and there's 2 choices of solution. There's just plain water, and then there's also a glucose solution. So with each cylinder, you're doing something different. There's one cylinder where you're filling the bag with sugar, sugar water, which is that glucose, and then you have the rest of the cylinder with just plain water. The second cylinder is having the water inside the dialysis tube and the sugar water on the outside, so it's just flipped and reverse. And then the third and fourth ones are control. Now, I don't want everyone to do the same control because that's quite a lot of equipment. So I will tell you, are you doing the water control or are you doing the glucose control? In these 2 ones, what is inside the dialysis bag and what is inside the cylinder is going to be the same thing. Thus, our control to kind of see what happens in those situations. What you're going to be looking at in this practical is how is the water moving? So is the water moving into the dialysis bag or moving out of the dialysis bag? The first thing we kind of need to know, though, is how do we set up those dialysis bags? Because they're a new bit of equipment that you guys haven't come across. So I'll play a video so you guys know how to kind of use it and tie it up. And then we'll talk about the actual specifics in our experiment. Hopefully there's sound.
I've started this experiment by first getting the dialysis tubing wet. This makes it much easier to actually pry it open so that we can place whatever liquid we need to inside of it. Initially, what you'll want to do is twist your dialysis tubing on one end and then tie that end off with a piece of string. Make sure it's a good snug fit for the knot so that no liquid can actually seep out of this end at any point in time during the experiment. Once the string is securely fastened and you're sure that no liquid will leak out of the bottom of the bag, then it's time to actually add whatever liquid it is you're going to add to it. So this is the first part of the lab where you're adding either a sucrose or water solution. Just simply pry the bag open on the opposite end, make a little bit of space so that the pipette's nose can actually dip into the bag. And then you'll take your pipette and fill it with whatever solution you want to add. In this particular instance, I'm using water. Fill up the pipette, and then you will place the nose of the pipette into the bag, press the vacuum release button, and slowly release the liquid into the bag. Be sure that you work the liquid into the bag so that the bottom part of the bag opens up. If not, the liquid's going to overflow and go out of the bag. So once you fill the bag up with the appropriate amount of liquid, you're then going to seal off the opposite end. So we will again twist the bag, and our goal here is to leave a small air bubble. We'll twist the bag, and if there's not enough air, then we'll move a little farther up. And once we've established it, we will twist it so there's at least a tiny air bubble left in the top of the bag. Once this is done, you'll take the second piece of string. You will wrap the second end of the bag in that piece of string and secure it tightly so that no liquid can leak out of this end of the bag either. Once you've securely fastened the string onto the bag, you should now have something that looks somewhat like a candy wrapper. We're going to use scissors, and we're actually going to trim off the string, the excess string, and we're going to trim off the excess pieces of dialysis tubing. This is just to make the bag a little easier to work with. Once we've done this, we're ready to take our bag and obtain its initial mass. If this is the exercise described wherein we place sucrose or water into the bag, we're going to need to first obtain its mass using the digital balance. So here I've teared out the digital balance, the large weigh boat on it, and I now place the bag and obtain the initial mass for that bag. At the very end of the exercise, we'll also obtain a final mass for the bag. After obtaining the mass of the bag, we're now free to place it in its respective beaker. Once placed into the beaker, the bag should be allowed to rest in the beaker for the appropriate amount of time as described in your lab manual.
Cool. Oh, that's 40 minutes later, so you can see what happens.
The purpose of experiment 6.2 was to determine the effect of solute concentration on osmosis. At the end of this experiment, you will once again obtain the mass of your bag using the digital balance, and depending upon the solution in which you placed your bag, you should either see an increase, a decrease, or no change in the mass of your bag. In experiment 6.3, you wanted to determine which molecules were able to permeate the bag and wind up in the beaker. At the end of this experiment, you will remove the bag from the beaker. There's no need to obtain its mass. Instead, you can simply throw it away. And then each group will come to each individual beaker.
So we're not doing that version. All right, so what I will do is I'll pull up the instructions for this practical and kind of walk you guys through it so you know what to expect. Cool. So let me show you where to find it. So this experiment is under lesson number 4, dialysis practice practical. We're going to be working on this all week. So for this first lesson, we should be setting up our dialysis experiment. First thing is you can find all the instructions for this experiment on the Google Slides. With that, keep in mind that you are allowed to do the first part in pairs, and you can only do it in pairs. So you're going to have a think about your partner, because I know we have 15, so there's going to be one group of 3. The reason why I want you guys to do it in pairs is because, well, it just helps with the workload. But also keep in mind that the pair that you are working with is the same pair that you're going to use for the surface area experiment that we do next term, and also the same person that you will be paired with for your actual practical. So do keep that in mind when you are thinking about who you want to be buddied up with, because it's going to be the buddy you're going to have throughout this standard. Cool. Anything in blue text I need you to delete and write. So in this case, you'll be deleting and writing the names. However, I want both people in the group to be working on their document because I'll be checking both, because there will be a moment where you guys will split up. Are we good so far? Yes, no? All right, so what I have done is I've given you guys this assess criteria so you're mindful of how you are going to be assessed for this topic. You guys can keep track on if you've completed something by writing in your checklist, and I will go through and write in red whether or not I have seen it or haven't seen it, so that way you guys can get some feedback. Be mindful that for the internal, we can use alternative evidence, and this will be an example of something that is alternative evidence that we can gather. So it's really important that you guys are completing this work and following the correct instructions. I have noted when I can't use it as evidence because we've provided you some guidelines because it is your first practical, and so I am scaffolding some of it for you. Are you okay with that so far? Ladies, are we listening?
Yeah.
All right, so that's kind of some things to keep in mind, that there's those guidelines in the assessment. The next thing on here is an introduction to explain what we're looking at for this experiment. So with this experiment, we're looking at osmosis, which is a form of diffusion. What we're going to be thinking about is high concentration of water versus low concentration of water. The other way you can think about it is high concentration of the sugar versus low concentration of the sugar. Now with this semi-permeable membrane, again, it's only going to allow certain things through that membrane. In this case, water. And so water is going to be moving in or out of that dialysis bag. So that's what we're going to be looking at in this experiment. I won't go through this in great detail and read it because we will cover it again later. The main thing for you guys to be mindful of is what is the aim of this experiment. So we're looking at, like I said, having a bag that has the glucose solution inside soaking in water. We have another bag with water that's soaking in the glucose solution, and then we have our controls, and I'll let you know which control to do. Cool, so you guys will need to fill in the hypothesis stuff. Now what I've done is I've put a colored box around things so you know what the requirement is. So if you see a red box, that is stuff that is going to the achieve level, so everyone needs to do that in order to pass. If you see things that have a blue box, then that's going up to the merit level, and anything in the green box goes up to the excellence level. So that just helps you kind of know what work needs to get completed. Like I said, we have provided you with the instructions, so you don't have to worry about them, but you would be expected to write your own method when you guys do future experiments. But since this is our first one that we're doing together, I'm providing you those instructions. Let me kind of walk you through some things that you are doing. So like I said, we have 3 different tubes that you're creating. Your dialysis bags will be living inside these larger measuring cylinders, and you are filling it so that way there is 50 mils of either water or glucose depending on which sample you have. And you have these little dialysis bags here, which have been soaking. And with these dialysis bags, you're going to be able to tie them up, fill it with the solution that you're supposed to be testing with, and then tie it to the other end. And then you're going to be putting them inside the beaker, and we're leaving it overnight to see if it absorbs the water or releases the water from the bag. Does that make sense so far? Yeah. All right. Cool. What other things that we need to do? So, I'll assign who has which control, but you're going to need 3 measuring cylinders. You're going to need to place one with 50 mils of the sugar solution, a second with 50 mils of water, and then the third one is going to be the control, and I'll let you know if you're doing sugar or water as your control. And make sure those are measured up to 50 mils. Then you need to prep your dialysis bags. Inside the dialysis bag, we are putting 15 milliliters of solution. And again, like I said, you're gonna have to like open it like you open a plastic bag. You're gonna have to just kind of do that, and then it'll open up at the side. So we need to make sure that we close off one end. You can twist it and tie it, or you can fold it over a few times and tie it. But you want to make sure that there's no potential leakage because that can affect your results. Then on the other end, you'll open it up and you'll put your 15 mils of solution. We do have beakers to make your life a little bit easier. And you also pipettes to make sure you can then fill it up. So you should have 15 milliliters of solution inside these little baggies. You're going to have a little bit of an air bubble, but you do want to try to get as much out as possible. The other thing to keep in mind is that you do want to keep some long string as a tail, so that way when you put it into the measuring cylinder, you can pull it back out later. Also be mindful that before you put any of the bags in their solutions, that you need to weigh them first to see what the weight is, because you're comparing the weight before and after. We do have scales, and I'll bring those out, and you guys can place it next to your station. But basically, you're weighing all three of them. Cool. And then what we'll do is we'll leave them on the benches, and we'll leave them there overnight. And then we'll check them tomorrow morning to see if they've gained or lost water. Cool. Some other things to note when you guys are doing this experiment, or what you're supposed to get done today, is I expect you guys to get your hypothesis slide number 6, number 7 completed. And also, number 10, you should be including an image of your diagram of what you've set up. And you won't have any results yet to, actually no, you would have some results. You guys should be inputting all your initial data. So there's 2 Google Sheets. There's one Google Sheet, which is your practical. And you should be noting the before and after volumes. And you should be also referring to the initial mass as well. Keep in mind that I've only created one of the tables for you, so you guys will need to create this second table as part of your task for today. So you should be recording the initial mass for the 3 ones that you have. And then we also have a Google Slides for us to collect class data as well. In this case, you won't need to worry about putting in the class data for today, because in class data, we want the % change in mass. Are we okay with that so far? Do people know what they're doing? All right, be mindful of your pairs, because you have to work with that pair throughout your assessment. Actually, we'll have everybody work in pairs, and then if the one person that's left over, we have an odd number, Mr. Hall can work with them to help, and then I can be walking around. Sound good? All right, so I'll put the PowerPoint back on, and you guys, I should say, fill in the slides number 6, 7, 10 on both you and your partner's Google Slides, and create your data table on the Google Sheets provided, and record your initial data. Cool, so that's the instruction for today. Anything else I need to tell you? I don't think so. We won't need goggles today because everything is just sugar and water, so you're fine. All right, cool, hop to it. I'm gonna check in to see who's in what group. Phoenix. Okay.